Remember Hitler does not do that. It's shameful. Yeah. This is very much right. Cool.
Yeah, right. Yeah. You know, I
Literally,
can't.
Can we talk about it now? No worries.
Oh, that's I mean, that's also better.
All right, sorry to push a little long to get started. That's the worst game
Describe the current tattoo.
All right, sorry to take a little long to get started. The instructor who's in here before me likes to turn off the computer. So this takes forever to get things fired up back up again. What are we going to do today? We're going to wrap up talking about this topic of horizontal gene transfer. And what I wanted to do today was actually do a bit of an exercise, and we'll talk about I think, pretty interesting case study, talking about hemorrhagic coli at the last bit of today, because I think getting through this lecture should be speedy, and hopefully this example, I think, helps synthesize some of what the major concepts of the last lectures have been. Before we get going today, are there any questions? All right. So I imagine many of you guys have started putting together your outlines using the collaborations that I've set up for you guys. One of the things you can do to let me know if you have kind of specific questions about items in your outlines is if you drop in comments, I get alerted of those comments, and I can go kind of address specific questions if you have them. Am I looking to give you general feedback? That general feedback will come with me looking through and grading your assignments. But if you have specific questions, I'm happy to give some preliminary feedback. Because ultimately, this assignment is worth a portion of what this project is worth. What's coming up is the big stuff, right? So I want to make sure that you're well equipped to begin putting together a nice, succinct visual presentation that takes us through essentially those questions that I've laid out for you in your outlines. So I hope you guys are all well underway. And yeah. No questions. No questions. All right. Making my job too easy, guys. All right. So what did we talk about last time? We ended our discussion beginning to talk about what makes a pathogen a pathogen. One of the mechanisms that drives, sort of molecular mechanisms that drives formation of pathogens is this process of horizontal gene transfer. and what we talked about last time, are getting a good way through these mechanisms. And then I want to kind of end today the formal lecture portion talking about these mobile genetic elements and pathogenicity islands. Essentially, that sort of packaging that we saw in the end of last lecture about how these virulence-associated genes are essentially in sort of discrete units that are found in prokaryotic genomes of pathogens. How does some of that happen, or at least what are some standing hypotheses about how this happens? And then giving some initial insights into how quickly something can become a pathogen. And we'll take a bit more of a look at that today with our case study. So there are three mechanisms, classical mechanisms that DNA is transmitted in prokaryotes. Prokaryotes, being haploid organisms, they do not reproduce sexually, and so they reproduce asexually by binary fission, and essentially are producing genetically identical progeny. And the only standing genetic variation that really begins to emerge is either through mutation or through movement of mobile DNA that we'll talk about later today. Now, where things get very murky and actually really erode this idea of there being a sort of biological species for prokaryotes are these processes of horizontal gene transfer. So essentially, large intercessions of regions of DNA. And when I say intercessions, it's essentially the regions of some other organism's chromosome or plasmid or some DNA that did not exist in that parent bacterium that now has become part of the genetic lineage of the offspring from this parent. So there are three mechanisms this happens by. We went through the first couple last time, so transformation, which is essentially some bacteria are capable of taking up DNA from the environment and incorporating that DNA into the chromosome. And that happens through basically neighboring cells dying, the DNA that's liberated out into the environment, that can be taken up by recipient cells that are naturally competent. And importantly, not all bacteria can do this. And it's a very specialized trait. But what's interesting is if we sort of reconstruct the ability, reconstruct bacterial phylogenies and look at the ability for competent transformation to occur in different lineages of very disparate lineages of bacteria, it's very clear that this is an old trait. So it's an old ancestral trait in the prokaryotic world, and that there are a couple of functional mechanisms that how this occurs. One in gram-positive bacteria, and a slightly analogous, but also very different mechanism in gram-negative bacteria. And the example that I gave you this last time really had to do more with the gram-negative mechanism. But regardless, both gram-positive and gram-positive and gram-negative bacteria, when they're taking DNA up from the environment, they have surface receptors that are binding to DNA and essentially using a molecular channel to take up DNA in cytoplasm. And the molecular motor that allows this to happen is essentially using the hydrolysis of one strand of the DNA to bring in the other. And what's interesting is that there's a fairly, well, I would say it's a near widely accepted hypothesis now that the ancestral function of doing this is actually to consume DNA, to eat it. And so, because a number of bacteria that don't actually engage in recombination of this single strand that ends up in the cytoplasm, they're able to eat the nucleotides that they're hydrolyzing using this DNA uptake mechanism. And so it's sort of, at least the hypothesis that seems to be most parsimonious, it's the simplest, sort of greediest hypothesis, is that this mechanism evolved to eat DNA, and that incorporation of the single strand of DNA was sort of a secondary event that happened while in the liver. But regardless, this is a very old trait in the bacterial world, that taking up DNA, incorporating it into your genome, this is a very old ancestral trait. And the mechanism that I gave you that's very common in gram-negative bacteria that engage in competent transformation is this modified type 2 secretion system. So it's actually, many of the components of this are really similar to that type 4 pili that we talked about, that retractile pili. So the outer membrane components of this system are essentially type 4 pili pieces. And the internal parts are sort of a hodgepodge of different homologous pieces from different types of systems that are essentially now adapted to take up DNA instead of certain proteins, for example. Okay, and these mechanisms are really important for shaping the evolution of a number of critically important environmental pathogens, so Vibrio cholera being one of them. So Vibrio cholera has an immense radiated diversity, and a large part of this is driven by the fact that they're able to take up DNA from dead neighboring Vibrio bacteria and incorporate fragments of those genomes into their own genomes. And so where this is problematic now, one of the later things, when we start talking about sort of mechanisms of evolution of pathogens, antibiotic resistance is one important thing. And so if you have a naturally competent and highly recombinogenic species of bacteria, if you have essentially sort of intercession into these lineages of antibiotic resistance genes, you can imagine that these get rapidly transmitted not only on plasmids but also chromosomally encoded regions as well. So transduction was essentially where we left off last time, and this is transfer of DNA, host chromosomal DNA from one donor host where this bacteriophage propagated itself, and delivering that DNA to a donor, a recipient cell or neighboring cell. And then what is happening here is that viruses they're good at packaging their own DNA, but no packaging process is 100% faithful. And so essentially any bacteriophage at some frequency, somewhere between 1 in 100 to 1 in 1,000 phage particles or viral particles will get packaged with host DNA. And when that happens, now these donor DNA containing viruses, we call these transducing particles. And so they're viral particles, they're viral packages that have DNA from the donor's chromosome that when delivered into a recipient cell, instead of there being injection of viral DNA into this recipient, it is injection of a region of chromosomal DNA from this original donor. And so this has found great utility in bacterial genetics. And so essentially, where, say, you're working in plants or you're working in some animal system, you'd be identifying mutations and you'd be doing sort of back crossing into a parental line and selecting for F1s, F2s, and segregants that are homozygous for a particular allele. What I would do in my lab is use generalized transduction, or this particular process, to move regions of chromosomes of mutants back into my sort of wild type original cells to test the effects of these mutations by themselves. So this has utility in the research world, but it is an important process that happens in natural populations because, yes?
I don't even interrupt you.
Yeah, no.
When I was doing research for our pathogen, I read in this one article it was talking about how bacteria, antibiotic resistance in similar species could be have a low likelihood of being transduced to this particular pathogen because it lacked homology. So does that mean that like this process is like really selective? So like that donor DNA is coming in all the time, but it's not often being recombined due to how similar the two genomes are.
Yes, and so that is a good point. So these first two mechanisms that I talked about are utterly reliant on homologous recombination to incorporate those things into the host genome. So transduction is a process that absolutely requires like high degrees of homology. So this is why you really only see sort of regions of DNA kind of moving around between closely related bacteria. Now, this process, while it helps if there is homology, it doesn't necessarily require it. So this is where you can end up with big integrations of foreign DNA into a chromosome, which is important for kind of how these pathogenicity variants move around, like we talked about. And so, or we'll talk about later on when we kind of touch on it. But the degree of homology between genomes is certainly a factor in how successful these transfer events are. But in natural populations, it is difficult to get a handle on how quickly these events happen, because usually the sort of output that we see, and we'll talk a little bit about this when we talk about the evolution of pathogens, is that essentially these things are happening all the time, and it just, there's such strong selection for a particular trait that if you are successful, you have clonal expansion of this one particular event that worked. And so the long story short, what we'll get to is that the evolution of pathogens is really sort of punctuated events where this sort of big innovation happens, and then there's this sort of standing variations, sort of these pathogens kind of middle along with sort of competing with each other because there's nothing new that's shown up. The pathogen acquires some new tool to interact with their host, and now its progeny are far more successful than the old.
So if you notice like a really common bacteria that picked up a new tool, would it be a useful application to, I guess, evaluate the level of homology between that pathogen and other common pathogens that are maybe commonly found concomitant with that one, to see the likelihood of that other pathogen developing the same new tool, essentially?
Yep. And so this is, thank you. No, we're gonna talk about these processes of how emergent and re-emergent pathogens. Some of the mechanisms that drive this are acquisition of things that didn't normally exist in this particular lineage of this pathogen. So yeah, and drug resistance is certainly one of the big ones because it's one of the hardest selection pressures. One of the, when I say hard, it's one of the most stringent selection pressures that we put on pathogens now as a result of what we do. Yeah, great question. Okay. Then I brought up this concept of lysogenic conversion, which is essentially when a bacteriophage, a temperate bacteriophage, will, through its own evolution, have acquired and sort of been kind of a garbage collector of genes. And this is something we'll talk about with mobile DNA in a little bit, is that these particular types of genetic elements, like viruses, they're just really good at just collecting genes. And in amongst ones that often get acquired into temperate phages are genes encoding toxins and other effector proteins. And you can kind of go back and look at that Salmonella example to illustrate that to yourself. But the most famous example of lysogenic conversion is actually in E. coli. And there's this Shiga toxin that is a AB5 AB toxin. Does some similar things to what it does some things similar to what cholera toxin does, and a few other things as well. Because there's a couple of different Shiga toxins that have slightly different active subunits. And so what was observed is that you can find pathogenic E. coli that harbor lysogenized, and so the temperate phages, either they undergo the lytic pathway, which is sort of standard viral replication, or they engage in this lysogenic pathway, which is integration of this phage genome into the host genome, where it just faithfully replicates along with the host genome until conditions such as DNA damage, severe physiological stress on the bacteria will result in induction of this lysogen into this lytic pathway where the virus begins replicating. So what was observed is you could take a spent culture supernatant of a lysogen or of a toxigenic E. coli, remove all of the cells, run this through a 0.22 micron filter, and take that sterile supernatant and basically expose your normal lab rat E. coli, K12 E. coli, to that supernatant, and you would end up converting your normal lab rat E. coli into a pathogen in one step. What happened was that essentially, at some frequency, you have phage induction all the time in large bacterial populations. And so in that spent growth medium from our toxigenic E. coli, there's some small amount of STX viral particles. They infected this otherwise like benign E. coli. And now that benign E. coli begins producing Shiga toxin under conditions of stress. And so if you think about the conditions that exist in your gastrointestinal system, they're not particularly super friendly to bacteria, especially in the midst of a active bacterial infection. If you remember the E. coli case, when there's inflammation, neutrophils and other innate immune cells start showing up and start producing reactive oxygen species. Reactive oxygen species will induce lysogen trend like activation into this lytic phase of viral replication. And when that happens, these cells that are now induced into this lytic pathway, in addition to viral replication, start overproducing Shiga toxin. And so this is part of what causes illness from pathogenic E. coli or certain pathogenic E. coli. And so one of the things that we end up not doing, and we'll talk about this in a little bit, is we don't treat with antibiotics if you have a say, hemorrhagic E. coli infection, because you essentially are just going to induce even more Shiga toxin production from these E. coli that you've been infected with, and you will not have a happy end if that's the case. And so what ends up happening, if you're diagnosed with a hemorrhagic E. coli infection, for example, you're going to get hooked up to an IV, you're going to sit in the hospital bed, and it's going to be essentially watching you and basically doing everything possible to help you and your body naturally clear such an infection. Okay, so the last and arguably the most prolific that happens in bacterial populations is this process known as conjugation. So this is essentially as close as prokaryotes are going to get to sexual reproduction. And what this is really a big driver of is transmission of plasmids. So many plasmids are self-mobilizable in the sense that they encode on them all of the genes required to make this conjugal mating bridge between two cells, often of the same species, although there are some self-transmissible plasmids that are capable of basically mediating very broad host range DNA transfer. And so, for example, there is a plasmid that that was isolated in E. coli long ago, RK2. And RK2, its transfer apparatus will pretty much deliver DNA into damn near anything with a membrane. So if that conjugal pilus, which is essentially a type 4 secretion system, if that is able to interact with a membrane, it will essentially transfer DNA into whatever that recipient is. So importantly, other things that can happen because of recombination of plasmids with the host chromosome is that those same self-transmissible mechanisms that exist for plasmids can also result in transfer of chromosomal DNA. And so the thing about this is that it's a very high efficiency process, and in the context of this class, I think it's most important to associate this with transmission of plasmids. And that sort of transmission of chromosomal fragments, this is something that happens really under laboratory conditions. And this is not something you frequently find in natural populations because there's significant fitness costs with becoming chromosomal integrant that's engaging in chromosomal transfer. So transmission of plasmids, where this is most important, the most famous self-transmissible plasmid is the F plasmid, the fertility factor from E. coli. And so basically this will transfer itself to non-F bearing E. coli very efficiently. And so this TRA region on this plasmid encodes a type 4 secretion system that ultimately creates a long retractile bridge into neighboring cells that don't produce this, that don't carry this F fertility factor or sex pilus. And what ends up happening is this pilus attaches to cells that don't bear F, essentially reels them in, and this type 4 secretion system essentially mediates formation of a double membrane spanning bridge, whereby this plasmid is able to be transferred as single-stranded DNA into a recipient cell, wherein this recipient cell, having this single-stranded DNA show up, begins replicating the lagging strand of this incoming piece of DNA, and as a result, you're immediately forming double-stranded DNA. This plasmid is recircularized by homologous recombination in this recipient, and now this recipient carries this plasmid. So this is the general mechanism by which all plasmids get transferred. And so feel free to take your time and kind of work through what's going on here, but essentially, the sex pilus is this type 4 secretion system, not to be confused with the type 4 pili that is a type 2 secretion system. I'm sorry I don't make up this nomenclature. But the sex pilus is a type 4 secretion system, and so what we know about type 4 secretion systems, it's interesting, is that they are able to transfer DNA. And this is what we commonly see them do because they're deeply associated with plasmid biology in prokaryotes, but they're also able to transfer proteins. And so what seems to be the ancestral function of this type 4 secretion system indeed is actually secretion of proteins, because there are some proteins that get secreted initially during this process, and beyond the scope of where I want to go in this discussion. But in some of the examples that I can't remember all of your pathogens, but there might be one or two, that there is a type 4 secretion system involved in translocating effector proteins. And so it's the same gig as transferring DNA. It's just that those type 4 secretion systems are involved in just delivering proteins and not DNA. And so finally, what happens when this happens is some really interesting stuff. So lysogenic conversion is one example where E. coli quickly become a really nasty pathogen through acquisition of a phage that allows it to produce a toxin. The same is true with plasmids. So plasmids, when you hear this, it's almost synonymous with antibiotic resistance. But in the context of this course, one of the things that is extraordinarily important for is transmission of pathogenic function. Okay? So for example, Shigella, which is more or less by comparison, like genomic comparison, is just E. coli. So it's E. coli that at some point in its evolutionary history acquired a single plasmid. On that plasmid, there is a type 3 secretion system that is integrally involved in invading epithelial cells and basically creating this vacuole. And once inside of the host, this same type 3 secretion system delivers more effector proteins that degrade that cytoplasmic, or that vacuolar membrane, and basically allow Shigella to be an intracellular pathogen. So we talked about how Shigella gets around using actin-based motility. So the way that it gets to where it's going is that it is unlike Salmonella. It doesn't enter epithelial cells through this apical side out in the lumen. What it does is it targets M cells, because M cells are naturally phagocytic, right? So they're busy taking things up, and Shigella get taken up by M cells. They are delivered to dendritic cells and macrophages. Essentially what they're able to do is be engulfed by these macrophages. Essentially they have all of the required equipment to withstand the low pH hydrogen peroxide production, radical oxygen species production that happens in these phagosomes in macrophages and macrophages and dendritic cells, and essentially are just able to escape these phagosomes. And when they do this, they're able to leave macrophages either via just sort of exocytosis, or that they kill these macrophages or dendritic cells. Then now they are down in this basolateral space down below these epithelial cells, and this is where their type 3 secretion system is integrally important in interacting with the basolateral side of these epithelial cells in your intestines, and they essentially invade from the bottom instead of from the top, like Listeria does. So they enter via this trigger mechanism. They're a vacuole. They lyse this vacuole, and then they start doing their nasty business in the cytoplasm of our epithelial cells. And so they keep moving from epithelial cell to epithelial cell via these cell-cell junctions. And in doing so, every cell that they enter, they essentially lyse this vacuolar membrane, induce pyroptosis in that cell, and just keep moving down through cell after cell after cell after cell. And every cell that they enter, that cell undergoes programmed death. And so you can imagine when this begins to happen in the epithelium in your intestines, this causes giant lesions in your intestines, and you ultimately end up with dysentery from this, which one of the root causes of dysentery is the erosion of the epithelial cell lining of your intestines. And so you essentially start leaking fluid into your intestines and blood and all kinds of stuff, and basically that ends up coming out, right? So that said, Shigella's ability to do this is all encoded in that plasmid. So if I take Shigella's plasmid and I put it into my sort of generic E. coli, things that it can't do, my generic E. coli doesn't have the toolkit to deal with macrophages. So that's some other things that Shigella acquired along the way, but it will invade the basolateral side of epithelial cells in tissue culture. And so that one plasmid acquisition event, essentially in one generation, like non-pathogenic E. coli has become a pathogen. Yep.
Since Shigella is a form, or I don't know if you would say a form of E. coli, is there any sense about like form sensing between Shigella and regular Klebsiella E. coli?
Yep. So they produce a similar kind of cadre of autoinducers, these acyl homosphenomactons. Yep. Okay. So who thinks that's cool? A quarter of you. So no one's impressed. Jeez. Okay. So before we head into this, are there questions? Do I have questions for you guys? It's good ones too. All right. No, I think this is a fun case study. Okay. So, how many of you guys have seen what I talked about before, minus the Shigella stuff? Okay. So these processes of DNA moving through bacterial populations fundamentally happen through ways. So in addition, there's another couple of elements that are at play in how pathogens are becoming pathogens. And so one important player is mobile DNA. And so essentially how these pathogenicity islands assemble themselves into pathogenicity islands is that undoubtedly when we look at these pathogenicity islands, they're often encased in mobile DNA, particularly transposons and integrated conjugated elements that we're going to talk about shortly. And so when I talk about mobile DNA, it is not DNA transfer. So this is not the same thing as horizontal gene transfer that we just talked about. So those are processes that are delivering DNA between organisms. Now, mobile DNA is essentially in the genome of our favorite bacteria. There are regions of DNA that will excise themselves and reinsert themselves into different places in a bacterial genome at fairly low frequency, but higher frequencies than what are typically observed with natural mutation rates. And so, what does this mean? Is that these processes are very common, and most of these mobile DNA elements that they move by a process known as transposition. And I talked a little bit about this in using transposons for mutagenizing things and understanding what pathogens do, but these transposons naturally exist in bacterial genomes. And one of the things that's important about these mobile DNA elements is that they frequently transpose themselves into transferable DNA. So when new DNA shows up, because like all of these recombinogenic functions are elevated in their activity, this is when transposons like to move. And so when you acquire a new plasmid, concomitantly, all of these mobile elements that you have in your genome, if you're a bacterium, have an increased frequency or increased probability of moving. So you end up with new DNA that shows up, and you end up with a transposon hop into, say, this plasmid, for example. And so when you look at plasmids, if I even go back and show you F, here, there's a whole bunch, at least these number of these IS elements and TN1000. All three of those things are essentially transposons. They're transposable elements that at some point, after moving through E. coli lineages, acquired these insertion sequences, or these are the old name for transposons, or this TN1000 transposon. And if we were to go and look at any natural plasmid in the bacterial population, is that they're going to be littered with transposons. And that's because of this sort of preferential movement of transposons when these horizontal gene transfer events are happening. So the two most common DNA elements found in prokaryotes are insertion sequences and transposons. And we'll talk a little bit about how these become much larger, more complicated things in a second. So insertion sequences, essentially what these are a transposon with basically no other genes. So they encode a transposase enzyme, and transposase binds to these inverted repeats on either side of it and induce catalytic excision of that piece of DNA and reinsertion of that piece of DNA somewhere else, random in the host gene, or onto a newly acquired plasmid. And transposons are essentially slightly more complex IS elements. So what transposons frequently are essentially this IS element had hopped and moved across some other chunk of DNA that often encodes, say, drug resistance genes. Because if you're going to, because there are a couple of different mechanisms that transposons move by. One is complete excision, reinsertion of these regions of DNA, and that's known as conservative transposition. So it's essentially cut and paste transposition, and then there's copy and paste transposition, which is essentially where movement of this transposon results in a duplication of a particular transposon. And most transposons begin their lives as sort of replicative transposons, and what will end up happening is you get duplication of this particular transposable element across regions of the chromosome, and through time and selection, usually one of these IS elements dies, so this transposase is no longer functional, and you end up with excision from these inverted repeats on the far ends, and essentially reinsertion of these transposable elements kind of all over the place as a result. So what's crazy about about transposons doing this is that they're hopping, moving, and you end up with things sort of captured in between these transposons. And there's lots of things that can get captured in between transposons. So antibiotic resistance genes are one, and the more and more that this sort of replicative transposition events happen, the more and more things kind of get acquired into this sort of nest of genes in between these inverted repeats. And you can end up with what are known as integrons that will carry sometimes upwards of 20 or 30 resistance genes sandwiched in a transposon. And even crazier is that there are a number of bacteriophages that have essentially, this is how they replicate now, is that they've been captured by a transposon, and that they use that transposon to replicate their genome. So instead of doing sort of circular replication of the chromosome, of the viral chromosome, they just basically induce massive amounts of replicative transposition. That's what a bacteriophage named Mu does. So they're less common than IS elements in bacterial genomes, but they're really important for shaping mobile content. So they're sort of really good at sort of sweeping in collections of beneficial genes, and then once genes are co-localized on the chromosome, essentially moving entire packages of genes or islands becomes a much easier thing to do than saying I have 30 different genes involved in making a type 3 secretion system that are dispersed in 30 different places in the bacterial genome. If that were true, probabilistically we would never ever see Shigella turn into or sort of lab rat E. coli turn into something that looks like Shigella in one quick horizontal transfer. Okay. So there are a couple of mechanisms that transposons move through. I kind of talked about this a bit. One is conservative transposition, so this is cut and paste, where transposon excises itself and pops into a new location. And then there's replicative transposition, where essentially this transposon is duplicated, so it's a copy and paste transposition. And so copy and paste transposition is often how IS elements sort of transform into transposons that begin to engage in conservative transposition. Okay. Going a little quick, because I think it is more fun to see the examples than some of the stuff that you've probably seen in the microbiology course. So, another example is enterotoxigenic E. coli that produces a heat-stable toxin, ST. And so this is actually contained on a transposon in ETEC, so enterotoxigenic E. coli. And so it's a very small toxin, 17 amino acids, so this is EPBE in terms of protein size, that interferes with host guanylate cyclases, or these G proteins, which are things that we saw Shiga toxin, right? Or cholera toxin. You're essentially ADP ribosylating those, and so you have uncontrolled guanylate cyclase activity, overproducing cyclic AMP or cyclic GMP, other cyclic nucleotides, and that induces sort of efflux of chloride anions, which begins driving water and sodium leakage from enterocytes. And so this is actually on transposon. And so kind of, depending on which toxigenic E. coli you find, it'll be in slightly different places on that host chromosome, but it's something that's preserved. And so heat label toxin is another toxin produced by ETEC, and it functions very similar to cholera toxin, similar to what the heat stable toxin does. So these HT and ST producing ETECs, these are commonly what's going to be associated with traveler's diarrhea. So if you were to say, go to Mexico, drink some unpurified water, and get horrible diarrhea, it's probably because of the combination function of these two. Okay. So these couple of examples of, say, Shigella and ETEC, they're kind of getting us at how small amounts of genetic information can get packaged, they can get moved. And in the case of plasmids, large bits can get moved around. But one of the bigger questions that's out there are you have these big tracts of genes, 50, 100, 200 kilobases worth of genetic information that are packaged together into these pathogenicity associated islands. And that these often have the ability to move between closely related species and sometimes very distantly related species. And this is a somewhat more kind of in the last decade innovation and understanding are what are known as integrative conjugative elements, or ICE, or ICE DNA. And what these do is that they are essentially conjugative plasmids that have sort of through passaging through different hosts acquired all kinds of different bits of mobile DNA, and they integrate themselves, they have integrated themselves into prophages, like Lambda. And so what these things end up being able to do is that they themselves are not really able to functionally replicate anymore, but they're able to mobilize this DNA. But bacteriophages, they're able to excise themselves and circularize themselves with their lambdoid or lambda-like phages. So what ends up happening is that these things get induced under lytic-inducing conditions, so things like production of reactive oxygen species, starvation, all kinds of things. They excise themselves, and then they get mobilized by trogenes, or like these type 4 secretion systems, into naive cells, naive recipients, and basically reintegrate themselves into these recipient cells. And so the hypothesis that's out there now is that these ice elements, if we kind of analyze these really big pathogenicity islands that are in, say, Salmonella, so SPY1, its origin was definitely one of these. It was an ice, piece of ice DNA that excised itself from some long ago ancestor of Salmonella, was transferred in, integrated, and became stably maintained in Salmonella's lineage through time. So are there any questions about this process? Yes?
The primary, I guess what makes this unique is just that it's much larger pieces of genetic information.
Yeah. And so unlike just transposons, which end up being sort of tens, well, we'll say thousands of base pairs in scale, now ice elements are essentially tens of thousands of base pairs in size. So you can end up with 100, 200,000 nucleotides of DNA that are using essentially bacteriophage functions for DNA excision. But now all of this stuff that was once upon a time on a plasmid is on the chromosome of a bacteria. Sort of, when things are integrated into the chromosome, they're much more stably maintained in the bacterial population than the plasmid. So plasmids, if I were to say take Shigella and grow it in my lab and streak it out from the freezer, for example, onto LB agar, and then I were to stain all of those colonies with the stain Congo red, I would see that there's a couple of proteins that are produced, surface proteins that are encoded on that virulence plasmid, that if I stain Shigella with Congo red, virulence plasmid containing Shigella will be dark red, otherwise they're sort of just white colonies like E. coli. So what I will see is if I streak out from the freezer like Shigella from my lab, is that, I don't know, maybe like 50% of the cells, 60% will maintain that plasmid, and the other 40% lose it very quickly. So there's no selective pressure to keep it, and so they lose it. And so this is where plasmid, like these virulence-associated plasmids, can get rapidly lost because essentially carrying accessory function that you don't need is something that can be quickly purged, especially for autonomously replicating DNA like plasmids. Whereas losing something on a chromosome is much more difficult to do. So, with that said, ice elements are thought to be sort of the origin of these much larger pathogenicity islands. So, based on what I've told you, do you guys think reconstructing the origin of a pathogenicity island is easy?
No.
No, right? But I mean, this is insane. So you have DNA moving around, hopping into other strange DNA that can excise itself. This is nuts. And so, sort of doing the molecular forensics to understand where certain things came from. This is extraordinarily difficult and is something that I think is a really exciting area of being able to sequence so many different pathogens from so many different places. Every outbreak that happens in the United States, isolates of those pathogens get sent to the CDC, they get typed, and they get sequenced now, and so there's just massive amounts of information that we can use to understand how these genomes are shaped, how these different functions are transmitted throughout the Ultimately, why care about understanding this? So this is one of the sort of gateways to understanding what are potential next pathogens, right? If you're going to try and forecast and say, we're doing this new thing, and we're always associated with these 5 bacteria now, and we didn't used to be. How might these 5 bacteria that we're now associating themselves with become nasty pathogens, right? So this is thinking about mobile DNA, horizontal transfer, and sort of understanding the origins of where some of these bigger packages of nasty tools for pathogens have come from that can tell us about what we might expect to happen in the future. Okay, so this brings us back to Salmonella. And so everything that I told you about these sort of transposable elements, lysogenic phages, ice DNA, all of this stuff has contributed to creating these pathogenicity islands in one of the most well-studied pathogens on Earth, Salmonella enterica type. So, with that, I'm happy to answer any questions. So I realized that was sort of a straight shot through that. Yep.
So you talked a lot about regular commensal bacteria becoming pathogenic and causing an infection.
Yep.
So when that's becoming virulent or causing infection, that's probably because of local DNA elements in the pathogenic version of it being transmitted to the regular commensal species. And then when you were talking earlier about things that the pathogen doesn't eat anymore that get eliminated, I guess like in future generations.
Yep.
Is that how it goes back? Like after you've been exposed to an infection, you're getting over it, and the pathogenic cells are killed off. Is that how it goes back to being commensal bacteria? Or?
So the frequency that say like enterotoxigenic E. coli is going to show up and transmit things to your native E. coli is really low. And so one of the things that works against horizontal transfer are a number of sort of self-defense mechanisms that bacteria have to just not acquire DNA. So it's not something that like you as a recipient, potential recipient, like acquiring DNA is just not something you're going to be super cool with.
What about with the mobile, like between species?
And so the examples that I've given about like these elements being able to move and quickly turn something into a pathogen, we've basically created like the most favorable conditions for that to happen in the lab, and we can get it to happen. Which indicates that that's likely how this originally happened. But Shigella transferring, say, its virulence plasmid into your commensal E. coli, the probability of that happening is very, very rare, and arguably so low that it just doesn't effectively happen. And one of the reasons is because of post-restriction modification. So many bacteria have the ability, so what they'll do is selectively methylate particular sequences on their genome. And so we use essentially the nuclease portions of these in recombinant DNA technology all the time. So we use restriction enzymes in any class. Like cleave specific, say 6 nucleotide sites of DNA. But that's something that many bacteria are using, but there's always an associated methylase that goes along with that restriction in the nuclease. That methylase is going around and methylating all of those 6 base pair sites. And if it detects that there's an unmethylated site, it cuts that DNA. And so by all of these mechanisms, sort of horizontal transfer, by any of the mechanisms that we talked about, that newly arrived DNA, one of those strands is always unmethylated. And so if there's one side of this thing that's unmethylated, these restriction endonucleases are going to come and cleave that DNA. So, additionally, CRISPR-Cas is another one that is essentially adaptive immunity for bacteria against horizontal transfer, a viral infection and plasmid acquisition. So if you've been infected with a particular virus or plasmid in your bacterium and you sort of survived exposure to that thing, oftentimes these fragments of these DNA get chopped up, sort of stored as molecular patterns that this CRISPR-Cas system can go and use to identify similar or identical DNA that shows up by a viral infection or plasmid transfer. Yeah. Those mechanisms appear to be sufficient to keep these horizontal events from happening in natural scenarios like your gut to a really good question. Right, so it looks like you guys are reasonably well distributed. If you're sitting by yourself, I encourage you to go with your friends, with your neighbors, because we are going to do a case study. So we're going to do hemorrhagic E. coli. So, hemorrhagic E. coli is something fun to study, but not to have. We'll talk a little bit about hemorrhagic E. coli again, but I think it's interesting to talk about hemorrhagic E. coli now. And what it is a form of E. coli that colonizes the colon. It's transmitted most frequently via contaminated food, and the most common source of hemorrhagic E. coli is through beef processing. So this particular brand of E. coli is oftentimes a commensal in cattle, and so the mechanisms that we're going to talk about, it doesn't do these things in cows, but it does them to us. And so hemorrhagic E. coli diverged from its sort of commensal E. coli ancestor about 4.5 to 5 million years ago. And so, is there anything interesting that happened 5 million years ago? A lot of years ago. So, like 6-7 million years ago, like when you had divergence of like sort of gorillas and stuff from these sort of from other apes. So 4 or 5 million years ago. So us, right? So unsurprisingly, sort of the evolutionary origin of this thing that still gives us bloody diarrhea seems to have shown up about the time that we started shit, right? Or at least many of our long ago evolutionary dead-end ancestors. So, what this thing does is it colonizes the colon, produces Shiga toxin, which is a toxin produced by Shigella. So it disrupts ion homeostasis, causes water leakage. In addition, it causes apoptosis of neighboring and infected cells. And so essentially this results in colonic bleeding and is really similar to dysentery in a lot of respects, except for reasons beyond what we're talking about here, you end up with just basically even more aggressive bloody diarrhea than you ever dysentery. So fun times, right? So if I were to see this, its clinical observations, I would see is that the patient would have bloody stool, and I would find very high titers of culturable E. coli that utilize sucrose. Now, what's interesting is most commensal E. coli do not do this. Okay, so sucrose is table sugar. There's a number of other things that hemorrhagic E. coli do that are sort of normal E. coli don't do. So they are urease positive. They'll eat sucrose. There's 1 or 2 other things that are not jumping out to me mind right now. But sucrose is one of the real big ones. And so you can screen them. You can isolate them and say McConkey agar, which is sort of a crystal violet bile salt lactose agar. So you can isolate them there, and then you can essentially test them as to whether or not they grow on sucrose. If they grow on sucrose, they'll probably be young if you have these other symptoms. So one of the freaky things that this particular pathogen does is, unfortunately, one of our patients died, and we want to understand what the heck this E. coli is doing. So we took a histological cross-section of their colon, and we started looking at the surface of epithelial cells in their colon, and we find these. These are E. coli cells. We use scanning electron microscope to see this, is that we have E. coli cells essentially sitting on a pedestal of host membrane. And one of your coworkers comes over and looks at these images that you're looking at, and you see thousands of these in this slice. And your coworker who came from a salmonella lab comes over and looks at this and says, "Hey, now this sort of looks like half-completed membrane ruffling, like with a normal endocytotic event that you see in cell. So, with that, you guys with this question. So what likely caused this cell surface peptide? A, that it is induced actin polymerization. Well, actually, I'll let you guys sit together, chat, and look at the responses. You guys can decide. I'll open this up, and you can
I
respond.
don't think it's right, because if it was accident polymerization, why would we place on the cell? We didn't have penetrated polymerase.
Yeah.
That would make sense because it doesn't have any, which I believe would likely be not.
Yes.
The triggers in there.
All right, so another minute or so, you guys can get your answers in.
None of the other ones in this.
I just thought that if it was acting polymerization, why is it really? Did he say that?
All right, so another 5 or 6 seconds to try to get our answers in. All right, you don't have them in? Sorry. All right, so what do you guys think?
Probably A.
Probably. All right, well, let's see what you guys think. A good number of you think A. You think B. So I laid out a hint that one of your coworkers thought was going on, and it had to do with something similar to what's going on with membrane ruffling that would normally result in endocytosis with salmonella. So this is actually a really similar process. So that there are effector proteins that are being delivered into the host that are inducing formation of this pedestal. Why EHAC and other related E. coli do this, why they do it, I couldn't tell you. But they do this, and it's interesting that they do. And how they do it is pretty cool. We'll see in just a second. So of all of these answers, the answer that is correct here is that actin polymerization in the host caused by the pathogen is what induced formation of this pedestal. Okay, which is another fun question. So given this is caused by actin polymerization, what is a likely mechanism? So effector protein delivered by a type 3 secretion system, needle complex, or binding to specific host surface receptors like the ECAD here.
A. You need to show them the effect. There's 5 all at once.
So I'll give you guys a minute for this one. What, 10 seconds to try to get the final answers in? Alright. Alright. So, most of the A's and 1 B. Alright. So, very technically, you can split the difference either way on this one. I haven't told you much else. I told you that it might be like salmonella. But both of these things would induce actin polymerization. Now, if this were a very salmonella-like mechanism, we'd expect a type 3 secretion system to be doing this dirty work. In either case, I just wanted you guys thinking about what possibilities could be. And so if we head down this type 3 secretion system road, so in this case, either one could be correct. What happens, actually, is that indeed a type 3 secretion system delivers effectors that modulate actin polymerization near this attachment site and cause dynamic changes in ion and water transport in cells that have been basically infected with these, or been intoxicated with these effector proteins. Now, this doesn't have a particular question, but I want you to think about this in that you've identified a potential adhesion involved in mediating this type interaction. It's a type 5 secretion system protein. Now you want to identify what the heck is this thing binding to on enterocytes. So you purify this extracellular portion of this adhesion, and you expose it to host enterocytes in tissue culture. And what you interestingly observe is that this protein fragment, that is this adhesion, only binds to host cells that have been previously exposed to EHAC. So what do you think that means? So what are kind of two things that might be happening? So do you think EHAC is inducing some new surface protein on the host that's produced by the host? Think it's possible? What's another alternative?
MHC2.
MHC2, potentially. So that'd be something derived from the host, right? So, what if the receptor for this particular bacterium was an effector protein? Right? So actually, that is what happens with EHAC and some of its relatives, is that they deliver all of these effector proteins, and one of them is a transmembrane protein receptor that gets integrated into the host cytoplasmic membrane, that EHAC produces its own adhesion for. So it's essentially like, well, you know what, I don't like anything that you produce to stick to, so I'm going to deliver my own receptor into the host cell and interact with that receptor. Now what's even crazier is that this effector protein recruits host cell proteins that some of these would be normally interacting with things like cadherin, things like these WASP proteins that are regulators of actin polymerization. They recruit this machinery to this receptor, and when intimin, the adhesin, interacts with TIR, this receptor adhesin pairing, that this interaction is one of the things that induces actin polymerization in host cells, so that essentially you have localized actin polymerization right underneath you, so you pop yourself up on this weird pedestal. And so exactly why EHEC does this, I couldn't tell you. But some of the other auxiliary things that happen because of this sort of artificial like host receptor being integrated is that this also interacts with a number of other host signaling pathways, like MAP kinase, a number of other things that modulate host cell behavior in very different ways. And so this is one of the, I think, the coolest examples of the sort of pathogen-host interface, where instead of trying to like deal with messing with host cell surface receptors to get the host to do what you want, what if you just delivered your own receptor into the host and that messed with the host, right? I think that's nuts. So, where I wanted to very quickly conclude, sorry I'm keeping you guys in the room, is that these things are, this entire business to do this is encoded in this LAE, which is essentially this locus of the parasite basement, and that this is a pathogen or CRE. So this is something that gets acquired and basically turns the already kind of pathogenic E. coli into a really nasty pathogenic E. coli that will stick around and hang out in the intestinal plasma. That is the case study for today.